how are you all? So today we are going to learn about neurons. You know what neurons are? They are the basic and the fundamental unit of your brains. Like we all know of the normal eukaryotic cell, right? These neurons are also similar to those eukaryotic cell, but they are little bit structurally and functionally different from that normal eukaryotic cell. So here today with me, you are going to learn about the basic characteristics of neurons and how they are different from the eukaryotic cell. Okay, so let's continue with the topic. This is how your neuron is going to look like. They are just a plexus. Like you can see a bundle and bundle of the nerves, they are joined together and they are united together and they are there to give you the signals. Like your brain is there to receive the signal, it is there to give you the signal, right? Then, what is neuron? Neuron is one of the basic cell of a brain. It is one of the individual cell. A nerve cell, right? This neuron, as you can see, it is having two types of projections, one from the top side and one from the bottom side. The one which you are watching on the top side, they are called dendrites, and one which you are watching on the bottom side, this is called axon. What is the difference between the dendrite or the axon? Dendrites are the one which are there to receive the signal, right? They are there to receive the signal from the body and they bring the signal towards the brain, right? What is the axon doing? It is the one who is taking away the signal from the neuron. It is taking away nerve signal, right? Okay, now, like I was saying, they are similar to the eukaryotic cell. Okay, so how they are similar to the eukaryotic cell? Let's first have a look on the normal eukaryotic cell. Of course, we are going to talk about the animal cell, right? Animal cell, on the outside, this consists of cell membrane. In the center, it is going to be the nucleus, right? Then, number of these projections on which ribosomes are present. This is called rough endoplasmic reticulum. Right? To this rough endoplasmic reticulum, of course, the ribosomes are attached. These ribosomes, they are going to form the proteins. In order to process the protein, Golgis are present. The Golgi complexes are present. To provide the energy, the mitochondria is present, right? So, now come back to the neurons. Here also, this part of the neuron, this is called cell body or soma or perikaryon. This cell body consists of number of granular cytoplasm. This granular cytoplasm, like your normal cell, is also having mitochondria, Golgi bodies, endoplasmic reticulum. Because here you can see that the nucleus is here, and as you have seen in the normal cell, nucleus is surrounded by endoplasmic reticulum and ribosomes for the processing of energy. So similarly here also we are having this endoplasmic reticulum plus ribosomes. You know what is the name to this? You must be confused that in the normal cell labeling you are never finding this ER. No beta, this endoplasmic reticulum, this is nothing. This is the same missile granules which you watch. These missile granules, they were discovered by Franz Missel, and yes, they are nothing different from the endoplasmic reticulum, and they are more present in the cell body or soma, and they are there to provide continuous protein synthesis, right? Now, let's proceed towards the bottom side, towards the axon side. This axon is starting from the base of the soma, and this part is called axon hillock. This part is called axon hillock, right? And what is the difference between the content of the cell body and the axon? Can you guess? Okay, there is no nucleus. I consider this point. Okay, in the axon, nucleus is absent. Nucleus is absent. What else is absent? Missile granules. Missile granules or your rough endoplasmic reticulum is absent. So can you guess more? When missile granule is absent, what else need to be absent from here? It is the Golgi bodies. Because we don't want the processing. Why we don't want the processing of protein? Because there is no manufacture of protein in this axon portion, because it was here in the cell body where the endoplasmic reticulum was present and Golgis were there to go for the processing. So we don't want the Golgi bodies also here, right? Because there is no endoplasmic reticulum manufacturing the protein, right? So what need to be present in order to have continuous energy? This is a cell, this is the part of the cell only. Mitochondria are present here. And what else? Some of the neuron fibers are present, right, in the cytoplasm of this axon. You know, what is the cytoplasm of axon called? The cytoplasm of axon, this is called exoplasm. The cytoplasm of the axon is called exoplasm. This axon, this exoplasm, this is surrounded by a sheath. This sheath is termed exolemma, right? This sheath is termed exolemma. You could have noticed the number of glia cells. What are glia cells? Give me a second, I'll be explaining you what we mean by glia cells. So you can notice the number of glia cells, calling here the Schwann cells in your peripheral nervous system. In your peripheral nervous system are surrounding the axon, right? And they are also then continuously covered by a layer. This layer is called neurilemma. Right? So this Schwann cells, and wherever the gap is present, this gap is called node of Ranvier. This gap is called node of Ranvier. This gap, this node of Ranvier, actually through which the nerve impulse they jump, and this jumping of impulse from one point of the node of Ranvier to the other point of node of Ranvier, this is called saltatory conduction of nerve impulse. What it is called? Saltatory conduction of nerve impulse. I hope the topic is getting clear to you. There is more difference from the dendrite and the axon. Like you can see in the dendrites, they were very thin fibers. The dendrites, they were very thin, and the nerve impulse was coming towards there. But at the end of axon, they are little bit thicker than the dendrite, and they have got some buttons. These buttons are called synaptic buttons, or synaptic vesicles are here, right? Synaptic knobs they are called. Synaptic buttons or knobs. Why? Because here neurotransmitters are present. At the end of this axon, some synaptic knobs are present, right? And they consist of neurotransmitters. These neurotransmitters are responsible for chemical transfer of nerve impulse, right? Then you have noticed that there can be number of neuron types. One famous type which you see, which is given in your textbooks, this is the multipolar neuron. What is a multipolar neuron? A multipolar neuron, where you can see a plexus of this, a lot of dendrites are there and axon is there, right? More number of dendrites are there and axon is also dividing. One end is receiving the signal, the other is taking out the signal. But in this pseudo-unipolar, you cannot notice the dendrites, just the two of the axon is dividing here. In the bipolar, it is having some dendrites and also it is having a part of axon. This you can see in your eyes. And in the unipolar, you can see that there is no dendrite present and more of the axon portion is present. This is most present in case of invertebrates. Right? So, at the end point, I want to tell you that these neurons, they are one of the basic fundamental cells of the nerve of your brain, right? But as you have seen, I have made the Schwann cell now. The Schwann cell is a part of peripheral nervous system. Then, what is present in the central nervous system? In place of Schwann cells, a special type of glia cells calling oligodendrocytes, they are present in the central nervous system, and they are there to provide the myelin. So now it is clear that the function of Schwann cell is to provide a layer of fatty tissue, and this layer of fatty tissue is called myelin sheath. So the neurons which are having the myelin coating, a layer of lipid coating, they are called myelinated or medulated, and those who don't have myelin, they are called non-myelinated or non-medulated. So like those who are having the myelinated neurons, they have got node of Ranvier, there is a faster transfer of nerve impulse. This type is called saltatory conduction of nerve impulse, and those who don't have myelinated neurons, they have a slower transfer of nerve impulse. Okay, I hope the topic is clear to everyone. Thank you.